Hello friends, I welcome all of you to our YouTube channel Pharmacy Student Resource. In this lecture session, let us discuss about receptor identification and preparation. First, let us see what is a receptor. Receptor is the catalytic region in an enzyme or responsive area in a protein like GPCRs, nuclear receptor, ion channels, etc. And basically, it is the empty space between protein folds in simple terms. Regarding any receptor pocket, we usually have three levels of information. Preliminary level, we'll have information regarding protein amino acid sequence or a gene sequence and its mechanism of action. Secondly, X-ray crystallography structure of only the protein. And third is the protein press co-crystallized ligand. What does it mean? Protein or gene or amino acid sequence is available, means the receptor is identified and its amino acid sequence is obtained and its mechanism of action is also known. So without having any other structural data. Here X-ray crystallography data is available, but we have absolutely no idea on active pocket or otherwise bioactive conformation of the ligand. In case of third one, protein plus co-crystallized ligand is there, means protein and its active pocket can be easily identified because ligand is also there. Bioactive conformation is also available to us. So this is the best data one can get regarding protein or a receptor. So this helps people to do computer aided drug designing easily or structure based drug designing easily. Now let us see at different levels with the data that we have, how can we prepare the receptor pocket and how can we identify the receptor pocket. Let us say when gene or amino acid sequence is available to us, then gene expression is done in any other species like E. coli or something like that, so that upon fermentation they will be getting protein in bulk. This they will take and they will develop an assay and confirm its catalytic activity or its role, biochemical role, and then they will also perform mutation studies. So point mutation is done to ascertain the amino acids and their role in bioactivity, and then they will try to identify inhibitors because an assay is developed. So in vitro analysis, in vitro assays were done or high throughput screening is done to identify the inhibitors. Then they will crystallize the protein or co-crystallize along with inhibitor and submit the sample for X-ray crystallography so that protein 3D structure is obtained. This gives us absolute data regarding protein. When protein could not be expressed, then other methods that are available is computational modeling like homology modeling. So here what they will do, they will take amino acid sequence or gene sequence and search it in the online databases. In case of protein sequence, BLAST is done. Then they will identify templates. By using templates, they will perform homology modeling, and through that a protein model is developed, and that is refined to ultimately obtain protein 3D structure. This is the best and most dependable way of obtaining protein three-dimensional data. Next, once the gene sequence is translated into receptor pocket, here you can see for we can easily obtain structural information regarding a protein. So where exactly rigid region is there, where exactly loop regions are there, and where exactly you have the catalytic site, everything is seen clearly once you get the three-dimensional structure of a protein. Then we can also have the receptor pocket definition, including what are all the various amino acids that are involved in the catalytic site. What exactly is the use of getting this data? So here this information helps us in defining the receptor pocket during structure based drug design. Here the catalytic amino acids whatever are there in the loop region are to be noted down separately in your observation book, so that this information you can use later on. Flexible or rigid. as you all knew, during catalytic activity of an enzyme or a response given by a protein, responsiveness of a protein is possible only when a protein is flexible. But complete flexibility, simulating complete flexibility needs a significant quantity of computational time. So that is the reason why we need to identify the amino acids that are there in the active loop, activation loop, or otherwise the catalytic site, so that we can define flexible residues present in the catalytic site. Now let us see how different ways are there to probe identification of a receptor and definition of a receptor. Basically we have two types of methods are available. One is topography of the protein is analyzed with the help of a rigid sphere, or we can call it as a point energy, and this kind of methodology you can see in case of Molegro virtual Docker. This is one of the example. The next one is grid based definition, means the entire region is kept in a grid box, and the search space is limited to only that grid box only. Along with this, other methods are also there where software can recognize ligand that is present in the active pocket, so that the region that is occupied by ligand can be defined as a receptor pocket. Or else we can also define a receptor pocket by identifying some of the amino acids that are present in catalytic loop, catalytic region. Now let us see how to prepare a file or identify a receptor pocket in different softwares. I'll give you a demonstration for a Molegro virtual Docker as well as AUTODOCK software. So this is Molegro virtual Docker user interface where this is let us input protein, because yesterday we have separated protein as well as ligand. In the last session you have seen that. Now I am downloading, or otherwise I am giving this. Okay, this is our protein. Now what is it that we need is the receptor pocket. How to identify receptor pocket? As I told you, it is now identification of cavities, means whatever empty spaces that are there between the protein folds which is accessible is actually analyzed. Here we have considered 5 angstrom units or 4 angstrom units as our the diameter of the sphere which we are using for probe. So if you reduce the diameter, we can go into the deeper regions in a protein. Yeah, these are the regions. These are the regions. You can see there are about 4 cavities were there. 4 cavities. 1, 2, 3, and 4. So in these 4 cavities, where exactly is your active pocket, that we need to see. Yeah, this is the ligand present in protein A, and the pocket over here is pocket number or cavity number 2. Cavity number 2. So while defining active pocket or during docking, maybe you will be able to identify. Okay, so the cavity number 2 can be considered as a region where docking can be performed. So this is the way how receptor pocket is defined in case of Molegro virtual Docker. So this is AUTODOCK tools. Now here let us see how to do the Yeah, here you have the green region is protein. The first thing we need to do is macromolecule, choose, we need to prepare the protein. Yes, it takes a bit time. Okay, okay, okay. Now your macromolecule is ready. What is it that we need to do is the grid point, receptor pocket, grid box. So grid box is the search space. Now you have to, because this is your active pocket where the ligand is also there, ligand. So this is the active pocket. Now how can we uh, define this box? You can move it. If you move this wheel, it will slowly come to the receptor. Okay, yes, now you have to see that it is completely entrapped. Good. Yes, now you can see that receptor pocket, this box is completely covering the ligand. Always you have to see that around this molecule at least 2 angstrom units width is allowed. A little bit of space is to be left around this molecule in order to, this is actually size, we are trimming it to see that if you define, if you give a lot of space, then if the search space increases, then time it takes for docking also increases, and many a times you may not be able to get appropriate result. So that is the reason why you need to do a little bit of work over here to define and redefine active pocket. Now what is it that we have here? The receptor pocket and its definition. What we need to note down is the center, X center 28.032, Y center 2.676, Z center 53.021. So this is the center, and dimensions X, Y, Z 56, 34, 30. So these are the things that we need to note down separately in your observation book. So this is the way how we define the receptor pocket. And when we need something like a flexible receptor, when you wanted to define a flexible receptor, because from the literature we will come to know about the amino acids that are present in our protein, as well as from the B factors we'll also know about the extent of flexibility of each of these amino acids. Hence, by using this data, we can identify the receptors, identify the amino acids, whatever are critical as well as flexible, so that we can identify them for defining flexible residues. Now let us see. Input, choose protein, select molecules. Yes. Choose torsion. Now you have to select. Let us say this is the amino acid. A. So whatever all the amino acids that we have over here, you can identify. Maybe let us say if this amino acid is very flexible, you identify that. Then another amino acid over here can also be identified. From the literature, the amino acids list can be obtained that are present in the active pocket. From the B factors, you can identify those amino acids which are highly flexible. So from then you can identify the receptor amino acids from this panel and then go back to flexible residues and choose torsions in the selected amino acids. It will take a little time. Then so it will tell you that how many rotatable bonds are there, and here you need to also see whether amide torsions are allowed, because amide bond is considered as non-rotatable for protein structure definition. In most of the cases it is non-rotatable. If you wish, you can activate this tab so that whether it is allowed or not allowed. Close. Flexible output, save as flexible PDB Qt. Flex. Okay, this is the way how receptor pocket can be defined. Okay friends, I hope you understood the way how a protein structure is obtained from the data that is available, and how to identify a catalytic region in the protein, and how to define a receptor pocket by using some of the softwares that are available. We'll continue our discussion with docking in the next lecture session. I thank all of you for your patient listening. If you have any queries or any comments, please post them in the comment box below, and we'll meet again in our next lecture session. Thank you all.